My name is Dana Philpott, and I'm a professor here in the Department of Immunology. My research interests are really mostly focused on inflammatory bowel disease. I have a real passion for trying to understand the pathogenesis of Crohn's disease. So we study innate immunity and host microbe interactions in the context of inflammatory bowel disease. So when I was doing my PhD, I was working on host bacteria interactions. We were studying the pathogenesis of a diarrhea causing bacterium called enteropathogenic E. coli. So I was always interested in how the host cell actually can detect these bacteria to respond and try to get rid of these infections. So then when I was doing my postdoctoral studies, in the late 90s, I'm not going to say what year, there was some seminal data that came out of the lab of Jules Hoffman from Strasbourg, showing how a toll receptor, which we know in humans called toll-like receptors, this toll receptor in Drosophila was involved in detecting both fungus, first of all, and then they also showed components of the system for bacteria. So that kind of the link between the mammalian host and the Drosophila host in terms of its ability to detect bacteria and other microbes really sparked my interest in this area, and that's where I am today. What this facility allows us to do is to study how the host can respond to microbes. So what we can do is we can take germ-free mice, so these are mice that are grown in isolated bubble cages. So these cages are hermetically sealed. They have air coming in through a HEPA filter. So these mice are completely devoid of any microbes. And then what we want to do with that is we want to then associate them with specific organisms. And that way we can really tell in detail what are the host cells responding to in terms of those bacteria or those bacterial metabolites. And in the long run, how these interactions then either lead to health, so we're interested in the interaction between bacteria and host in terms of health, but also how these microbes can then affect different diseases. So this is what we call the robo-gut. These are actually chemostat cultures of bacteria that we grow ex vivo. So what we do is we take a microbiota from, we either have patients' human samples of poop or mouse samples of poop, and we grow out the different microbes from these hosts, and then we grow them in these robo-guts. So this simulates the gut environment, these culture conditions. It's all anaerobic and balanced in terms of pH. We have this constant input of food going into these cultures that contains all sorts of yummy things, including mucus, to grow these bacteria that are specific for the gut. So what we can do with these is that we can grow different cultures of bacteria ex vivo. So these are bacteria, again, that are strictly anaerobic. They would die in oxygen. And then once these communities equilibrize, we can then put those into mice and, again, test whether these bacteria have an influence on the type of disease that we're interested in looking at.
My project focuses on looking at the interaction between the immune system cells in your intestines and how they interact with the different microbiota in your intestines. So other than bacteria, which you hear about a lot, there's a whole lot of other microorganisms such as yeast and fungi and parasites that are in the gut. And I specifically have been studying on a specific parasite or protozoa called Trichomonas mirus and how it interacts with immune cells. And this is really important because it can alter your susceptibility to developing inflammatory bowel diseases such as Crohn's disease, which is a big focus in our laboratory. So one really important piece of advice I'd give to any grad student is make sure you look for a lab that's very supportive, a supervisor who's going to be there for you and support you, colleagues in your lab who are going to help you out and like talk through problems with you, and also to make sure that you have a really good support group outside of your lab too, with your peers and friends outside, because that's really what's going to help get you through graduate school and really make the experience for you is more the people who are there to help you and also just to relax and have fun.
Hi, my name is Dave Prescott. I am a postdoctoral fellow in Dana Philpott's lab at the University of Toronto in the Department of Immunology. I am studying the microbial sensor NOD2, of which the gene for mutations are known to be the most strongly associated genetic risk for the development of the inflammatory bowel disease, Crohn's disease. And we know that this gene is involved in the sensing of bacterial products, but we don't know how this leads to the development of overt inflammation. So my project specifically is looking at this gene in specific tissues and mouse models and trying to identify novel functions for the gene in cell types maybe that have not been addressed or assessed in the past. The hours, while you end up working a lot, sometimes it seems like almost all the time, you really get to make your own and there's a lot of flexibility. And it's actually a lot of fun because every day you have the chance of discovering something new or finding something that nobody has ever seen before, and that's really one of the only places you ever get that opportunity.